I'm very happy to introduce today's speaker, Dr. John Ashton from the University of Rochester. I've known John as a friend and as a colleague for the last 15 years since he was a graduate student. John did his bachelor's from the Rochester Institute of Technology, followed by a PhD at University of Rochester. Uh, and he has continued working there, rising through the ranks, initially starting as instructor and is currently associate professor. Uh, John has also got an MBA from the Simon School of Business at the University of Rochester, which is one of the leading business schools in the country. He has been very successful academically as well as a scientific leader, and he played a vital role in the expansion of the genomic research center at the University of Rochester, and handles its functioning, its finances, as well as its expansion. He's got several publications, several grants, and we are extremely happy that he is here presenting today. So once again, John, thank you for taking your time to do this.
Thank you, Sid, and thanks for the invite. I'm really thrilled to be here, and thanks to Amy for helping me navigate the Zoom virtual thing. So I appreciate that. I've known Sid for a long time, so it's really great to see him in this capacity, and hopefully I don't disappoint him with this talk. So today I'm here in my hat of director of the Genomics Research Center. So you can see that I do a lot of different things at University of Rochester and at Wilmot. But today we're just going to focus on some of the evaluation and expansion of omics platforms that we've been working towards here at Wilmot, as well as other cancer centers in the country. And just give you a little bit of unpublished data on some of the comparisons that we're doing in the single cell space. And then I hope to end with trying to give you a sense for the complexity of cancer research and most biomedical research, but also to give you an understanding of how we all are thinking of trying to integrate those types of data sources and really get a holistic approach to not only research, but also potentially diagnostic medicine. So, as we all know, the central dogma of most modern biology is you have DNA that makes RNA that makes protein. We all know that that's more complex than it is in that linear form. But really, for many, many years, folks have studied the genome and tried to understand mutations and mutational status, and using that for not only research information, but also to create diagnostic markers for many different diseases. And as we all learn now, there's more complexity to that, and more diagnostic companies as well as research are moving into integrating not just mutational status, but RNA transcript expression and various forms of RNA biology, as well as spatial temporal dynamics. So, and using more spatial omics methodologies, using the proteome, and really trying to get a full sense of what's going on mechanistically. So today I'm just going to focus on the majority of the talk around the transcriptome and giving you a little bit of flavor for some of the tools that are out there and how we've been trying to basically wade through all of the particular methods out there and evaluate what might work the best for specifically cancer research with human patient samples. Then I'm going to just touch briefly on some of the newer proteome analyses that really reduce the barrier for this type of effort in cancer research. And then at the end, I'm just going to give you a very broad perspective on where I feel the field is and how complex it is and how you have to integrate all of these components together to really be able to understand not only diagnostically, but also from a research discovery perspective on what you need to understand and know to really understand the mechanisms behind biology that will help infer what kind of treatment paradigms there might be available and or where the field might want to go in the future towards developing those therapeutics. So this is all really driven by high throughput sequencing in the last 10 or 15 years. For those of you that don't know what high throughput sequencing really is, I've just tried to articulate that here. Basically, hopefully everyone can see my pointer. Basically, the simple concept is you have a sample, whether it's tissue, cells, or a fragment of a tissue from a slide. You isolate nucleic acid, you make some sort of cDNA or DNA library, and you use a number of different sequencers that are available in the market to generate sequence level data. then the real challenge is analyzing those data. But for the most part, this is very standardized now. There are many different sequencing modalities out there. There are a lot of good data analysis packages that make this not that make this less of a barrier and more of a an easy, accessible thing to the majority of the research community. In addition, what really helps drive this is not only the sequencing technology, but the cost. So today we always say that sequencing has been commoditized, and that means the cost for sequencing has dropped dramatically. As you can see over the last 20, 15 years, the cost has dramatically decreased and continues to decrease. And this really is driven by the technological revolution and the desire for some market competition between different sequencing vendors. So today, as of now, with the new sequencers, you can do a human whole genome, 30 to 60x coverage in less than a day. And this is also buttressed by not only generating the data very fast at a cost-efficient scale, but also with the advent of AI, we can analyze data much, much faster. And we can use AI and machine learning to help identify some putative results and observations that we can then go validate in the lab. So I don't want to make it sound so simplistic. Again, there are many different ways of doing this. In the clinic, I'm sure you're used to getting tissue and being able to assess tissue not only morphologically, but also with DNA-based tests or sequencing-based tests. Um, how you actually go about understanding the DNA mutational status, the proteomics, transcriptomics, really depends on multiple different assays that you have. Again, most of what is being done in the field these days is sequencing, but contrary to popular belief, microarrays are making a comeback, and in certain data generation types, microarrays are very, very important, particularly if you're looking at epigenetic um, results. There are microarrays specifically designed that work really well for looking at epigenetic status um, of certain CPG islands that have impact on disease state. But the point of this slide is really to show it is very complex, and I want to make that very clear. And we're going to focus right now a lot on the single cell transcriptomic space because this is something that is evolving over time. It's now becoming more of a steady state assay and technology that both research and clinically are being used. And I think it's important to kind of understand the landscape. So just to give you a little primer for those of you that may not be completely familiar with what I'm talking about, historically, sequencing has all been developed around something we call bulk RNA sequencing. So in essence, this is a really good analogy. You basically take tissue or cells and you make some homogenate of them, right? What you end up doing is you're able to look broadly at the transcriptional expression in that particular, in this case, smoothie, but you lose the context. You don't understand where those genes might be active in what cell types. And in some cases, you might lose information because you get dilution of a rare subtype, for example, with a very specific signature. If there's only 5 cells in millions and millions of cells, as you can imagine, that would be diluted out and it would just become part of the background and you would lose that granularity. So that's where single cell helps a lot, where instead of it just being a homogenized smoothie, you're able to then look and break apart each of those cells individually to be able to characterize what type of fruit they are, for example. Right? And that's great because you can specifically identify rare subtypes. You can look at different cell characteristics. If you're interested in a complex tissue, like with cancer, in most cases, you're looking for those tumor cells and you want to understand the signals that are intrinsic to those tumor cells, but you also want to understand how those cells might be influencing other cells around them. So single cell is great to be able to say intrinsically what's going on within specific cells, but it doesn't help with that last piece of how are cells communicating with each other. And so now we've evolved into spatial transcriptomics as a field, and this is really where it doesn't replace bulk RNA sequencing or single cell necessarily, but it gives you more context. Not only does it allow you to identify specific cells, it allows you to identify where those cells are, what their activity is, and how other cells in their niche or their in microenvironment are impacting their growth, their response to chemotherapy, and how they're bidirectionally impacting them to do potentially niche remodeling, and to provide a more protective environment for cancer cells, for example. But like I said before, we're going to just focus on single cell RNA sequencing for the moment because I think it's sufficiently complex. And there's a lot of effort in the field to use these types of methods in diagnostic reporting. And so just to give you a little bit of a different way of looking at it, and for those of you that might like to have a little bit more technical detail, this is just a schematic, really trying to peel back the onion on my smoothie and fruit analogy. Really where bulk RNA sequencing is, as you can imagine, heterogeneous tissue, you lyse everything, you have the whole repertoire of RNA, you sequence, and you get bulk expression data. So you get very broad expression. And this is where cellular heterogeneity is masked. You aren't able to see what's specifically going on in a single cell, let alone a subset of those cells, like B cells or T cells. And single cell sequencing, as we talked about, is really isolating each of those cells independently and just within those cells looking at the RNA content. And that allows you to be able to identify specific cell clusters based on the transcriptional state and look at differential expression and to see what transcriptional signatures may be changing over time or as a consequence of disease burden or chemotherapy, for example. And this really helps reveal that heterogeneity and gives you the power to look at cell populations. So, for example, a lot of my research is focused on acute myeloid leukemia, and the field has long known that what drives that disease are a very, very rare subtype of cells that are present at a very low frequency. And as you can imagine, if we're trying to understand what drives that specific disease, bulk RNA sequencing can only help us so much. We really have to be able to break apart the cell heterogeneity to focus on those specific that initiating cell compartment. Otherwise, we may not have enough information to understand how we can fully push that disease into remission or complete remission where we've targeted that small population that drives that disease. Single cell has evolved a lot, and this is just to give you a sense of back in early 2000s where everything was with a manual process all the way to where we are now. And you can see there's been a lot of iterations of technology that have really iterated around these four or five, six different technologies. Most of the methodologies that are out there now still do nano droplets and a version of well-based methodologies. But you can see there's a lot of technologies that are out there, even in 2017. And the question for us as a research center studying cancer is what is the best or the best group of, assays or technologies that will help us answer the majority of the questions for cancer research. So currently, there are three main players that are out in the field, and I'm showing them here: 10x Genomics, Parse Biosciences, and Illumina, who recently acquired a technology called Fluent. They're all very, very robust in their own right. But for us, as a cancer center studying cancer research, we want to be able to identify what is the best method for individuals and what are some of the other methods that may have specific use cases for our researchers here at Wilmot Cancer Center to help empower them. So really briefly, 10x Genomics is, like I mentioned before, this is the nano droplet-based methodology where you have single cells that you pass through an aperture, and then at a single cell level, you get the cell to bind with a hydrogel bead that has all the reagents for the reaction, and an oil droplet. At the end, you'll have a cell encapsulated by itself with all the reagents that you need. You do this with thousands and thousands of cells. You collect those cells at the end. Each of them will then go through a process that allows them to be barcoded so that you identify the particular cell in that droplet. And then I'm not going to go into too much of the detail, but in the end, you're able to sequence that and identify the transcripts that came from that specific cell. So this is great because it's very low, it's very high throughput. You can do thousands of these reactions in a single pass. And this is really the gold standard for the last probably 4 or 5 years. The majority of folks that do single cell use 10x Genomics. Recently, Parse Biosciences released a different type of methodology. It's really based on a lot of work that has gone out in the field from Stanford and Boston and Cornell, around a process called combinatorial indexing. So this is where you basically take cells. In this case, you fix the cells first to allow permeabilization of those cells so that all the reagents will go and flow into a cell. You take those fixed cells and you go through this process of basically putting them on a plate and then sequentially additional plates that at the end, each of these wells have a unique barcode. So in the end of this sequential process, you have individual cells that have a unique barcoded code to them, so to speak. And you can see here an example of that where there's different colors of different barcodes, and you use the combination of those barcodes from sequencing to identify the cell of interest. And then finally, Illumina, I mentioned Fluent, bought this company, which really makes this super easy. You basically take your cells, you put them in a tube. They have a bunch of polymers in this tube. You vortex them. And then, like with 10x, you generate these little nanospheres around a single cell. And within that cell, again, you have oil and you have a barcoded hydrogel template. So very similar to 10x, but without the need for any special instrumentation other than a vortexer. And in the end, you get this emulsion that looks like this. So each of these nano droplets contain a single cell. And the benefit from this is it's very easy. It's very easy for any resident or clinical person late at night that's working on a surgery resection of a tumor. They can easily take the tumor, dissociate it, put it in this tube, vortex it, and get to a stable stopping point. And so for us, when we're evaluating these technologies, the benefit for Parse is that we can fix cells. So again, thinking of a clinician or a resident in the middle of the night trying to collect for a research study, there's a lot of benefit and value to being able to fix cells and then give them to the genomics center here to be able to finish processing. And equally, there's a lot of benefit from research labs and the same kind of principle of residents that are doing research late at night or on the weekend to be able to make this really easy and efficient for them to take tissue, dissociate it, and do something with it. So for us, these two pieces are really meant to try to expand the access to individuals here to be able to do single cell studies. This is just to give you a little bit a sense of, I don't want to spend too much time on this, but really in the end, price point is important to most people. And you can see that the per sample price varies drastically between all these methodologies. And again, the price by itself is just one part of the equation. How many cells you can get, what type of profiling can you do you need to fix cells or not? Or can these particular technologies be amenable to fixation? And so this is the space that we're in, and they offer a lot of value to the research community that we're evaluating. So just to give you a primer, these are the different types of assays we tested for each of these technologies. And these are the labels that, keep in mind as I go through the data, you'll have to remember because these are the labels that will be on those plots. So 10x has three different assays we tested, Parse one, and Illumina was one. So I alluded to before that my research is very much focused on acute myeloid leukemia. So we decided here there have been a lot of studies that have been out in the field trying to compare these methods as well as other methods. And they generally use either cell lines or not very complex or hard to access patient tissues. And so a lot of the comparisons are more technical. How well does this assay work? But we really wanted to understand how well these current technologies that we're going to evaluate really perform in a real life use case setting. And so again, we decided to use AML as the use case. And for those of you that are unfamiliar with AML, it's a hematological malignancy. It really affects the bone marrow of individuals. And the way it works is that there's a hematopoietic stem cell compartment and progenitor compartment that acquires mutations over time that causes a shunting of normal hematopoiesis into the normal immune cells that we all need for immunity and just health into these leukemic progenitors that really create a differentiation block of end-stage cells, and you get these things called leukemic blasts. So you basically remove the normal hematopoietic cells and you go into this leukemic blast stage. That really is where you get an overproduction of these cells that can not only impact bone marrow, but also other organs as these cells will leave the bone marrow and cause other issues in different organs. So this is where we wanted to test because at the end, we're able to look at a lot of heterogeneous space, both in looking at rare subtypes, looking at normal cells that are within that AML sampling, as well as the leukemic blast. So it's a really heterogeneous population that has a lot of impact and research value to us in Wilmot because one of our primary cancers that we study is hematological malignancies, AML being one of them. So we went about doing this design experiment where um, we acquired patient-derived AML apheresis samples, and we chose two AMLs, two different, both of them having a different percentage of blast content. So AML1 was a low blast patient sample, and AML2 was a high blast. Again, trying to understand the heterogeneous space where if we have a lot of blast cells, that's going to be the majority of the single cells we see, and we wanted to ask throughout these technologies how well could they perform in a somewhat more homogeneous space? And can we then see those rare populations that might be present in that with that context? And then the other extreme being low blast count, we should still have a lot of normal hematopoietic cells, and some cells that are basically state transitioning between normal and malignancy. So we did this again based on the technologies. We acquired these two AMLs. We did fresh versus fixed because as you remember, parse is a fixation protocol. We targeted 5,000 cells for each of the comparisons. And I'm just showing down here the actual data that we were to identify. So again, GemX, OCM, and Flex are all the 10x genomics methods. And you can see for each AML how many cells we actually were able to not only theoretically target, but actually target for each of these technologies. And then we decided to sequence. And so the other question that we wanted to ask is, what is the real impact of sequencing depth? And so, oops, sorry. Most of the vendors require 20,000 reads per cell. So that's 20,000 data points per cell is their recommendation. But we wanted to ask because we have some internal data from a lot of the single cell work that we're doing that depth matters. And so if you sequence more, what will be the value in more genes detected and what might that impact be? So we wanted to ask for all these technologies, is there a saturation point where is 20,000 reads per cell sufficient? And if you do anything higher than that, what is your return on that investment? Because it will cost more. So preliminary conclusions, like I mentioned, the platforms perform similarly overall, with some exceptions, like I just showed you with Illumina, that we're still trying to tease apart the real reasons behind that. And all platforms that we tested, again, the three that we tested were Parse, 10x, and Illumina, are viable options, and that the selection really needs to be dependent on the cost, accessibility, and ease of use. And in particular, once we really flesh out what's going on with Illumina, whether or not that is a true biological phenomenon, that might allow Illumina to have a bigger stake in the game of for rare populations that might have low gene detection, at least from our data, that suggests that Illumina would perform better in those situations. Um, just briefly, again, there's a lot more work that we're doing with this. And the biggest thing that we're trying to work towards now is cell typing to understand whether or not the Illumina artifact that we see is truly an artifact, or if we're able to really see specific cell types that we know are present in these AML samples. Some of the other things that we're hoping to do as well with these data is to really explore the value of long read sequencing. Most of what we've done here today that I've showed you is with the most predominant high throughput sequencing methodology, which is short read, which most of the vendors support short read. But the question of how each of these methods would allow us to do long read sequencing to give investigators the research data pretty much open-ended. They might be interested in looking at BCR, TCR, repertoire. Um, if you're using short read sequencing, you have to really optimize the workflow for that purpose, whereas long read will allow people to capture all of that data at full transcript length, and also allow them to look at splicing variation as well as translocations, which are technically hard from short read sequencing. And so we're going to use these data and some of these um, libraries from this experiment that we generated to really test that question of what's the value of long read sequencing? It is more expensive, and to see how each of those methods might be able to perform with a long read data set versus short read. And importantly, most of these methods are now coming out with multi-omic capabilities. I mentioned earlier at the beginning that it's not just about RNA, it's not just about DNA anymore, it's about everything. Then being able at a single cell level to not only look at DNA mutational status, transcriptional status, but also protein is really, really important. Some of these methods do that better than others, but that's a separate study for a different day. And I just wanted to provide a use case example of what we've been doing here at Wilmot using single cell and bulk RNA. This publication came out back in May in Nature. It was a collaboration between myself and a good colleague of mine here, Dr. Javisha Bajaj. Basically, again, I mentioned that we're very much into hematological malignancies. So Javisha and I have a longstanding history in animal model of AML. Um, and so what we undertook here was to ask the question of, can single cell help us with a temporal analysis of how, when you initiate leukemia, it models the microenvironment and what particular cell types are important for the longevity of leukemia as well as the initiation of leukemia. So basically, this mouse model, very briefly, is we have a syngeneic mouse model where we add two mutations, BCR-ABL and NUP98-HOXA9, into a C57 black 6. And this experiment was really designed that over a series of 2 weeks, because this disease is very, very rapid in this animal model, that we took several time points. And the point of this was to look at early versus initiation of leukemia, and then expansion, and then end stage disease. So to really try to mirror the pathological course of what we see with human AML. And then we basically digested the bone marrow and separate out the leukemia cells from the bone marrow microenvironment. And then from the bone marrow microenvironment, we performed single cell analyses on each of those time points. We were able to identify a number of cell types within the bone marrow microenvironment, and different states of these different cell types. Like you see here, arterial 1, 2, and 3, they're different states of the same cell type. Then we were able to also characterize how those cells change over time, and really be able to see proportionally how certain cell types like osteo1 drop over time as disease burden increases, and how other cell types increase over time, which was great. We're able to see in an animal model over time how the disease remodels the microenvironment and what cells specifically are being targeted or impacted by that. Then we were able to further go on and show not only what those cell types that are going up or down in proportion, but also what are some of the pathways and signals that are being modulated in those populations that could give us some clues as to what the leukemic cells are specifically doing to, for example, impact MSCs, endothelial cells, or these osteoblast cells that are being reduced over time in the leukemic development. So then we use that information and some primary bulk RNA-seq data from human patient samples to basically understand and create basically a niche interaction map where we understood now the genes that were being impacted from these cells, and we had the tumor cells, leukemia cells that we separated, that we could look at what are the genes that they're transcribing and be able to come up with a network of, over time, the leukemia cells transcriptional signatures are changing. These are the genes that are being activated. We know these genes are secretory or are local interactions. We know what their ligands are. Can we map out that interaction space? And so a lot of what this paper did was be able to identify and characterize these niche interactions, to be able to create a more a characterized atlas of leukemia initiation and progression, not only within leukemia, but within the microenvironment to get a more holistic picture. And this was great. This really showcases how single cell technologies can help do this, because if you imagine trying to do this from bulk RNA-seq, it would be impossible. Which is great. So from a basic research perspective, that's awesome. We can do a lot of stuff with single cell and a lot of stuff with other methodologies, but can we do more? And so instead of just the basic science approach where we want to mechanistically characterize, some pathology, we wanted here at Wilmot to try to see, can we push the envelope and can we help support our population science and our epidemiology colleagues that really do a lot of human health work, and a lot of this is moving into mobile blood collection, mobile health. where they're asking patients from remote sites to collect blood themselves and send them back to the institution to do something. As you can imagine, you can't get as much blood from these types of mobile blood collection devices. So we really set out to assess ways that we can help support our cancer prevention and control colleagues, in particular here at Wilmot, in these types of avenues. So as I mentioned before, sequencing has been commoditized. So now there are high throughput methodologies that we can use for proteome analyses. Olink is one of them. It's a great example of one. There are other ones as well, but our goal here was to say, do we have a way that we can generate data that are focused on inflammatory cytokines for our CPC colleagues that can be done from 4 microliters of serum or blood, doesn't require a huge price point, and doesn't require complex analysis, right? You can imagine if you're trying to do exploratory analyses, mass spectrometry might be the best platform to use. Very laborious, very cost prohibitive, and a lot of places don't really offer the level of sensitivity that you would need for 4 microliters of volume, for example. Um, and there are other methods, but basically we chose to evaluate Olink. It's a recent panel that was developed by Olink and released. It's 1,000 or so proteins, and we can do medium to high throughput with this. So the goal of this was we want to support mobile blood collection. Is this a platform that we can use in order to be able to support profiling immune inflammation cytokines? I won't spend too much time on this other than this is all antibody based, and so it's for those of you that are familiar with ELISAs and ELISA sandwiches, it's a very similar type of technology, except for they use DNA oligos to cause a bridging effect when two antibodies bind to a specific protein that allow us to then make a DNA library and put it through the same process I showed you earlier for NGS. The most important part of this for us was the quality control, and not only the internal quality control that Olink provided, but also the plate controls. So if you want to do a large study and you have a 96 well plate, you might need 10 of those plates. The value here is that there are internal plate controls that allow us to leverage that for larger studies. And so there were a lot of reasons why we chose to start with this. Probably the biggest reason is this goes right into our normal workflow. This is very similar to what I showed you before. It goes right into our pipelines and workflows for bulk RNA sequencing. So it makes it really easy for us as a genomics sequencing center to be able to process these data and really turn out the same type of results that folks are used to. And those are shown on the right. For just context, because I'm going to be showing some data, Olink works a little bit differently than most other methodologies. So they normalize all of the expression into this normalized protein expression matrix called MPX. And that's with some of the internal controls on each plate. This is how they look at relative abundance. This is not an absolute abundance measure. And it's all in the log2 scale. And so for most of us probably know that what a log2 scale means, but just for context, again, a 2 to the 1 is 2x of the control, again, relative to the controls. I'll quote through this quickly just to give you a sense. I mentioned we wanted to test this method because this will allow us to use the data in a very similar way that we're used to. And this just shows an example of that. So a lot of the data and the reports that we can generate are very much what we're used to from the transcriptomic space. And so it was a really easy lift to be able to move the data into this analysis space, both for QC control and principal component analyses, as well as how the data looks, right? We're very used to looking at box plots for samples and variants of expression. And we're very used to generating heat maps and understanding what those heat maps mean and being able to help interpret those data for investigators. And as well, same thing with volcano plots and other types of things. So again, the point of this is that this would look very much like RNA sequencing analysis if I had not told you that it was all protein based. Pathway enrichment, again, it's something that is an interesting component, but again, it's a very targeted panel. So the value of pathway analyses when it's a biased panel for immune cytokines, you can argue the validity or the helpfulness of that. So I just want to finish up briefly with a little bit of a perspective on cancer research, as most research is a multi-omic problem, right? We no longer just look at genomics. We no longer just look at transcriptomics. A lot of this is an integrative approach looking at the multitude of different omic spaces. Those of you are probably aware that a lot of the clinical diagnostic companies like Tempus, Caris, Nativa, and other ones are moving their clinical diagnostic infrastructure, if they haven't already, to incorporate genomics, transcriptomics, and single-cell transcriptomics, and even proteomics. And some are actually moving into spatial omics and metabolomics as a way of adding data to increase the clinical utility of what they're reporting out. And this is true on research as well, that, if not more so, to be able to understand the real underpinnings of any disease biology, you have to be able to integrate all these different data types to get the fullest picture, to be able to really understand what's driving malignancy or any type of disease, but also what you could potentially do to intervene. And I'll leave you with the newest era that we're in is spatial transcriptomics and spatial genomics. There's many different ways of doing it. This is just giving you an example of the whole concept is that you have a tissue slice and you're able to identify cells and interrogate cells within their own milieu, within their own microenvironment, and able to look at transcript expression, potentially mutational expression, clonality, all those types of things, but in a spatial context. This doesn't replace single cell assays or bulk assays necessarily, because single cell data is really used to infer more information that allows you to then go into spatial omics methods and from what you learn from single cell, really extend your knowledge. And this is the hope, right? Is the hope that you'll be able to reconstruct a heart, for example, based on spatial and single cell data that you can look at not only the patterns of expression over time, subcellular structures to look at different pieces of cells that are different internal components of cells that are important for biology, cell-to-cell interaction more directly than what I showed you in that publication, and really be able to look at the morphology over time and be able to add that to the mix of data on how normal development or disease biology might be abrogated by certain morphological constraints. And with that, I'll end. I just wanted to mention that all of that data that I showed you and the work was done by the Genomics Research Center here at U of R. And they're part of a department of advanced research technologies. These are the lab members and the team that we have. And that they're a really strong team. And this is a mix of bioinformatics folks and wet lab folks, but none of what I showed you could be done without their work. And with that, I'm done and happy to take any questions.
Thank you, John. That was fantastic. I'm going to be meeting separately with Dr. Ashton after this talk, but and I have several questions, but I encourage others. Yes, Dr. Scott Dem.
Hey, John, that was great. Thank you. Thank you very much. With regard to the last couple of slides focusing on integration of the multi-omics with the spatial omics, how is your core in the Cancer Center and more broadly at University of Rochester organizing this initiative? I've been very impressed with your core and how you've organized your epigenetics profiling and your DNA sequencing and then the services that you provide. So I'm really interested to hear your vision for how to provide this broadly across your institution.
Yeah, no, that's a great question. Thanks for that. So I think it's a challenge at most places, but we're working here towards creating infrastructure to support this. So part of the issue is trying to understand algorithms and data modeling methods that will work well for this, which we're actively doing. The other piece of this is that we're really leveraging University of Rochester as part of in New York State. We have this Empire AI Consortia. So it's a group of other institutions that all have AI and machine learning interest. So we're really leveraging that to help extend and create tools to enable people to do this, as well as research collaborations to identify better methods in order to integrate those data. So it's really a institutional slash multi-institutional effort. And part of at least my role here in Wilma is to help the institution create the infrastructure in order to support that. And so a lot of my time is spent aligning investigators with a data science interest with, biomedical researchers. And part of that is also, we have an R25 training grant that I'm on that helps support graduate students in data science and bringing them into the fold to help towards that research. It's more complicated than that. There's a lot of investment in certain areas that we're working towards, and we're kind of doing this in real time in an iterative fashion. But learning from other institutions within the Empire AI Consortium has been a big help.
Great, thank you.
Sure.
Dr. Crossan?
Yeah, thanks very much. I mean, this is just an expansion of Scott's question. And it has to do with basic medical education. As you know, the medical education for the medical students is actually changing. And obviously, the technologies you talked about today have tremendous diagnostic capabilities across all of medicine. To what extent do you think the current medical school students coming out of medical school understand these capabilities?
Great question. Um, so I can only speak for our institution. Um, I can give you broader perspective, which I think is probably similar, but not as much as they need. And so they do get some exposure, at least in our medical school. We have curricula that span some of the research departments, and so some of those folks have been able to expose medical students in this space. But over the last year, we've been pushing to have more of this type of broad content into the curricula. For example, in December, I'll be presenting to the medical students on a couple lecture series of this type of technology, at a level that they can kind of understand. I don't expect them to understand all the details, but to get some more exposure to this. And then that sets up other seminars and things that we work with to kind of give some more extended support. So the goal of that is for me to come in, give that exposure, and then we have a very robust training initiative within the Genomics Center here, where for years we've been hosting workshops and seminars at different levels of complexity that we all host through our research library that we then give them access to, and hope that they self-align to some of those workshops for those that are interested. Some of those medical students are MSTP folks that will also do research. But I do think, to your point, the fields are evolving very quickly, and I think it's on all medical schools to try to enhance their curricula such that the students are at least exposed to some of this, because I believe 100% that spatial omics, spatial transcriptomics, or any genomics will eventually logically go into a pathology level assessment, right? If they can leverage that and know about that, I think that's better for everybody.
Thank you.
Well, if there are no more questions, once again, John, thank you for a wonderful talk. And I'll be meeting with you at in the next 20 minutes around 9:15 Central. So thanks again. Thank you, everybody.
Thank you, everyone.